What I'm going to do is really give you a quick tour around antibody incompatible transplantation and why it's important in the wider context of transplantation and some of the things that we've been talking about in terms of tolerance, complement, and so on. Really, this is all I want to tell you, so if you've got that, you can probably go for lunch now. There's a difference between HLA and ABO. The antibodies actually aren't that different, but the antigens are different. And HLA incompatible and blood group incompatible transplants are very different. And one of the things that I think is worth asking is, why is that? These transplants are technically the same. You put a kidney in, you do the anastomosis, you take the clamps off and it works, but immunologically they're very difficult to do. And if we can do these transplants, then we can probably do most other transplants. And therefore I think we may have a lot of lessons to learn from these types of transplants. So blood group incompatible transplants, we know, when we talk about blood group antigens, in fact, these antigens don't just appear on red cells. So we're not really worried about the blood that we're putting in when we transplant a kidney. We're worried about those antigens that appear on the cells of the kidney or the pancreas or the liver or the heart, and they all actually appear to a different extent in different organs. Kidney is one of the hardest to do. The antibodies to these are in fact antibodies to gut bacteria, which happen to cross-react with these antigens. And we know that from this paper, JCI in the 60s, where they took a bunch of children and fed them cups of E. coli and saw that their blood group antibodies rose. Some of them had to stop and be sick and have some diarrhea, and then they carried on the experiment and showed that you could induce blood group antibodies. So what we know in the UK, and this varies according to different countries, if you're O phenotype, you'll have anti-A and anti-B antibody. If you're A, you'll have anti-B, and so on. So you can see that the most common blood group incompatibility that we're faced with is A into O. If we think about HLA incompatibility, there are three reasons why you might have HLA antibodies. So most of you walking around will not have HLA antibody in your circulation to any significant extent. If you had a previous transplant, pregnancy, or a blood transfusion. And the figures here are just really markers for the degree of sensitization. And you can see that it increases with the type of sensitization. Indeed, once you get onto a transplant plus a blood transfusion, so if Martin Drage is operating, this is highly likely, you'll get a very, I'm joking, you'll get a very high rate of sensitization. And interestingly, these antibodies are not to the blood, they're actually to the kidney. There's some confusion about terminology. You'll hear people talk about PRA, that's probably outdated now. What we're using in the UK is calculated reaction frequency, and you'll see that in some of the slides. What that means is if you take the last 10,000 deceased donors in the UK, what percentage of those do you have HLA antibody to? So if you have a calculated reaction frequency of 99%, you're not going to get a deceased donor transplant because you've had antibody to nearly all of the last 10,000 donors in the UK. Okay? There are degrees of sensitization, simply Luminex positive, so bead positive, flow cross-match positive, and CDC positive. These represent strong degrees of sensitization. And we, in our unit, don't really think of Luminex positive donor-specific antibody on its own as being particularly important. There's some debate about whether it increases the rate of graft loss, but we don't think, generally speaking, it's a big problem. So we're really concentrating on the flow cross-match or the CDC positive cross-match. So how common is the problem? It's about 20 to 30%. So 20-30% of the patients coming forward for transplantation will have either HLA antibody or will have a blood group incompatible donor. So it's a big problem, and that's probably going to get bigger as time goes on and we're retransplanting more and more people. About 25% of our living donor program at Guy's is antibody incompatible.